So here we go. The first outline begins with just an overview of the urinary system and then a little bit on kidney anatomy before getting into the functions. So here is a good diagram from the textbook showing all of the major structures that are part of the urinary system. So I'll give you just kind of like a quick description of each one. We have two kidneys, which are here and here. And these are the organs that filter blood to produce urine. And then coming off of each kidney, you can see on the diagram the yellow tube that extends down from each kidney. These are called ureters. And these are tubes. Again, there's two of these. They're tubes that transport urine from the kidneys to the bladder, which is located right here. Okay, and then the urinary bladder, which is what I just pointed out, the ureters lead down to that. This is a storage area for urine. And then finally, the urethra, which is not really pictured on here. It's kind of hidden by other structures here, but this is the urethra. It is a single tube that carries urine from the bladder to the outside of the body. Okay, and then for the functions of the urinary system, these are listed in your outline already, so nothing to fill in here. But one thing that I talked about with the lab group that I met with is that the kidneys are often thought of as just producing urine, and that's it. Usually that's the main function that you think about when you think of the kidneys. But really that's a secondary function that is a result of their primary function. And the primary function is to regulate, basically regulate blood and body fluids. It's regulating what is in your blood, the composition of your blood and body fluids. So it regulates the water content of blood, so that therefore influences blood volume, which has a direct effect on blood pressure. It also regulates the ions, the solutes, like the solid particles that are in blood. So ions, nutrients, also substances that would influence the pH of blood. And also kidneys produce several different hormones as well as an enzyme. And then here's the secondary function as a result of doing all of that. So any excess water, excess ions get excreted by the kidneys, and they're leaving the body through urine. So that is the excretion of waste. All right, and we'll talk more about the functions of the kidneys in a little bit and how they perform that function. But first, we're going to do a little bit more of the anatomy for the kidneys. So here's the location. The kidneys are actually pretty far back in the body. If you're looking at this cut here, so if you cut the body like in a transverse section, which goes this way, and you look down into the abdominopelvic cavity, this is the front of the body, and this is the back of the body. You can see the vertebral column right here. The kidneys are right there. So they're like on either side of the spine. They're pretty far back. I am going to skip this part right here, blood supply to the kidneys. You don't need to know all of these arteries, mainly just know that blood going into each kidney comes through the renal artery. That's the main blood vessel that brings blood into each kidney. And then the vessel that drains blood out is called the renal vein. And we'll get into these a little bit, but there's a capillary bed that's located within the kidneys, as well as smaller arteries called arterioles that send blood into this capillary bed and drain blood out of that capillary bed. So now I want to go through just some of the external and internal anatomy related to the kidney. So here, this is actually the exact picture that's in your lab guide that you'll see when you do the kidney dissection, or that you saw if you did it already. So this is an actual diagram of the kidney, and this is a real kidney from a cadaver. So you can see what it will kind of look like when you do the dissection. There is a sort of like a depression on the medial side. That's like the inside of each of the kidneys, and that's called the hilus. So the hilus is the depression, or sort of like an indentation on the medial side. And that's where the ureter and these blood vessels, the renal artery and the renal vein, go in and out of the kidneys. Okay, and then surrounding the kidneys, I don't have a picture of this, but there's 3 layers of tissue that surround the kidneys. A lot of it is for protection. The first one that's listed on your outline is the renal fascia. This is the outer layer, outermost layer. It's like pretty dense connective tissue. And this is the layer that keeps the kidneys attached to the abdominal wall. So this is the layer that's kind of connecting the kidneys to the body wall and keeping them held in their place, so they're not just floating around in the abdominopelvic cavity. All right, that's the outermost layer. And then the next one listed on your outline is the adipose capsule. And if you remember from A&P1, adipose tissue is fatty tissue. It stores fat. This is the middle layer, which is adipose tissue. So it's filled with fat. And for that reason, it protects the kidney. It adds just some cushioning around the kidney, so it's protected from physical trauma. Okay, and then the last one listed is called the renal capsule. This is the innermost layer. And when you do the dissection, this is the only layer that you will actually be able to see because it directly covers the kidney. It doesn't come off. So the renal capsule it's a very thin, transparent membrane, and it actually helps protect the kidneys from infection. Okay, so next on the outline is that blood flow picture, the same one right here. We just talked about mainly the renal artery, renal vein, and then the arterioles and capillaries. So the vessels that are in the boxes, if you're looking at the outline, the couple on the top, and then the arteriole and the capillary beds that are listed on the bottom, those are the only ones that you would need to know or recognize the names of. All of those arteries and veins listed in the middle, you don't need to know, and we won't even ever mention them again. But they are just smaller arteries and veins that are part of the network that bring blood either into the kidney if it's an artery or out of the kidney if it's a vein. Actually, I'm going to leave this picture up for a little bit longer because now we have the internal anatomy of the kidney. So this is what I'll show you guys. We have a model of the kidney that looks exactly like this. So I'll show this to you. I showed it to half of you, and the other half will see it when you come in on Tuesday for the dissection. There are 2 main regions within the kidney. The external region is called the renal cortex. That's the outer portion. And just one term listed underneath of that are the renal columns. These are extensions of the cortex that they extend in between the pyramids, which we'll talk about those in just a second. So these are the renal columns are extensions of the cortex, which go or extend in between each of these pyramids. So the cortex is this lighter colored tissue that you can see if you're looking at the kidney diagram right here. The renal medulla is the inner portion of the kidney here. And there are several structures that are located in here. This is the bigger region, as you can see. So there's more stuff in this area. And the thing that's probably the most obvious are the renal pyramids, which are these triangular structures. They're dark, this darker color. One thing that you can't see associated with these pyramids are the next thing listed, renal papilla. And these are small openings or holes at the end or at the tip of each pyramid. So what we'll discuss eventually is the formation of urine. And these holes are actually to allow urine to pass through. So urine is going to come out of those little holes, which, like I said, they're so small you can't really see here, but they're there at the end of each pyramid. And then from there, urine is going to go into a series of channels, which are the next structures that are listed. So these channels are called calyces. The next thing on your list is a minor calyx, which is a channel leading off of a renal pyramid. And then a major calyx, basically several minor calyces empty into a major calyx. So the major calyx is a larger channel that several minor calyces, which are these smaller channels, empty into. Okay, so the channels that come directly off of each pyramid here and here and here and here, these are minor calyces. And then like right here, where several minor calyces come together and unite, they form a larger channel. That's called a major calyx. All right, then here you have just like an open area where all of the calyces, where they all empty into, and that is called the renal pelvis. So this is a sort of a large region that all calyces empty into. It's right in the middle. Okay, and then one more thing is the renal sinus. A sinus is always going to be like an opening or a space, and that's exactly what this is. The renal sinus is the space or the cavity that contains the renal pelvis and all of these calyces, all of these channels. So if you were to remove these channels and the renal pelvis right here, this entire area right here in the middle, and you can actually do that when you do the dissection, you can kind of scoop them out almost. If you take them out, you'll see that there's like a cavity there. There's a space there, and that is the renal sinus. So it's just the location of all these channels and the renal pelvis. And their job is really just to carry urine. And urine is going through these channels from the smaller ones to the bigger ones. Then it gets into the renal pelvis. And then finally, once it's there, it can leave the kidney through the ureter. Remember, the ureters are the tubes that exit each of the kidneys right here. You can see part of it here. So that's the order of the flow of urine after it's produced. Okay, so next thing to talk about is the tiny structures that are located inside of the kidneys, which are microscopic. These, and one is pictured right here. These are called nephrons. These are tiny, tiny microscopic structures that are within each of your kidneys. There are millions of these within each kidney. And let me see, you guys have a definition for the nephron already in your outline. This is really like the main functional unit of the kidney, because this is what is actually filtering blood to produce filtrate, which ultimately becomes urine. So it's performing the job of the kidneys. As far as the location for the nephrons, these are found primarily in the renal cortex. Remember, that's the outer region of the kidney. And they do extend down into the renal pyramids. So they're primarily in the cortex, but then they do extend down into the renal pyramids. One thing that's pretty interesting about these nephrons is that you are, the number that you're born with is the number that you are going to have for your lifetime. You don't make more of these. And unfortunately, if they are damaged, they can't be repaired, and your body can't just make more. So if they're damaged, they stay that way. And if a lot of them are damaged, ultimately it's going to affect kidney function. Okay, there's two parts to the nephron. The renal corpuscle is this first section, and the renal tubule is basically the long tube that kind of coils around and forms the rest of the nephron. And then the function of the nephron is to filter blood, to produce a fluid that is called filtrate. Okay, and that filtrate eventually becomes urine. Okay, so we will talk a little bit more about the nephron on the next like page or two of the outline. And that goes into nephron anatomy. So we're just going to start with the renal corpuscle, that first part of the nephron, and kind of work our way down. So this is what a typical nephron looks like. And like I said, this first section here is called the renal corpuscle. It's made up of the glomerulus and the glomerular capsule. So first thing is the glomerulus. That is the red stuff, if you look closely, that you can see inside here. And when I say red stuff, it actually is, it's like a mass of capillaries. So it's red blood vessels because they're carrying oxygenated blood. So it's a mass of capillaries. And they are specialized for filtration. These capillaries perform filtration. Okay, a couple of details about the capillaries, which are in your outline already. They are fenestrated. That means that they have little holes in them. They have little like spaces, gaps in their walls. So that makes them porous. It means that substances can get through their walls. Another thing is that if you remember Actually, I don't know if oh no, we did. We talked about blood pressure in the various blood vessels and how typically it's the highest in arteries, and then it drops once blood gets into the capillaries, it drops by a lot. And then by the time it gets into the veins, blood pressure is really low. So in these capillaries, however, in the all these glomeruli, blood pressure actually is higher than it is in normal capillary beds. But that actually serves a purpose. That's a good thing here, because this blood pressure is actually helping to force substances across the filtration membrane. And that's what you want to have happen here. Filtration of blood is happening here. You want that to happen. So that extra blood pressure is helping to push substances through the filtration membrane. And this is the only capillary bed in the body that is supplied and drained by an arteriole. Usually capillary beds are supplied by an arteriole, so that's what brings blood into it. But then coming out, blood is drained out by a venule, which is like a small vein. So this is kind of unique here because you have an arteriole actually draining blood out too. The other part of the renal corpuscle is the glomerular capsule. And this is a cup-shaped structure that surrounds the glomerulus. So it's the yellow colored structure that surrounds the capillary bed that you see on the inside. So it surrounds that like this. And there are two layers of the glomerular capsule. There is a parietal layer, which is the outer layer or the outer wall, and a visceral layer. That's the inner layer that directly touches the glomerulus. So here is a sort of a better or an enlarged, at least, picture of the glomerular capsule and the glomerulus. Here is the glomerulus. So that's the mass of capillaries that you see here. This is the glomerular capsule. So you can see the outer wall, but it curves around. And what you might not be able to tell is then it does extend all along the outside here of the glomerulus. It covers these capillaries. And then it comes back around the outside again. And then you do have a space in the middle in between those two layers. So just a little bit more about that inner layer, the visceral layer of the glomerular capsule. And I think this was mentioned, actually, I'm pretty sure it was, in the urinary physiology lab that you guys did last week. So this layer is composed of cells that are called podocytes. You can actually see that here. This is one of those cells. And these cells contain pedicels, which are thin extensions. That's what you're seeing right here. It's thin extensions that wrap around the capillaries. So it's almost like fingers. If you look at these extensions, these pedicels, it's almost like fingers that wrap around the capillary like this. So in between each of those extensions, you can see there's a little gap. Those are called filtration slits. So those are thin spaces between the pedicels. Okay, and those gaps, like those spaces, are what allow substances to pass through. This actually, and we'll talk about this later, but this forms part of the filtration membrane. So those spaces allow substances to pass out of blood and get into the nephron. And I did mention in between those two layers of the capsule is the capsular space. So you can see there is a space there. And this space fills with filtrate after filtration has happened and blood or substances from within blood pass through the filtration membrane. This is where they go. So now that stuff is called filtrate once it enters the capsular space right here. Okay, so we covered just now the glomerulus and the glomerular capsule. That's this area right here. And then the rest of the nephron is this long tube. It's called the renal tubule. So the renal corpuscle, those first two parts, that's where filtration happens. It's in the renal tubule that the other two processes involved in urine formation occur. So those processes are called reabsorption and secretion. So it's throughout the entire renal tubule that those other two processes are taking place. Okay, so I'll go through the parts. There's four other parts, four specific parts of the renal tubule. Right here, the proximal convoluted tubule, and that's a long name, so we just abbreviate that PCT. And then down here, this looks like a loop. It's called the loop of Henle. And then here is the distal convoluted tubule. We just call that the DCT. And then the last section that goes all the way down, straight down, is called the collecting duct. Okay, so back to the beginning, back to the PCT. This is the first part of the renal tubule. And for histology, this isn't really that big of a deal, except you should know that it's made of just like simple cuboidal epithelium, like little cube-shaped cells. But what's significant is that they contain microvilli. Do you guys remember villi and microvilli from the digestive system?
Almost like little fingers.
Exactly. Mm-hmm. So they are on like the surface of these cells. And does anyone remember what they help with? Like why you have
absorption?
Yes, exactly. So that's what they do here too. They increase the surface area available for absorption. That's exactly what they're doing here. They increase the surface area for absorption. In the kidney, it's called reabsorption because you've actually already absorbed these substances within the digestive tract, and then they went into the blood. Now, substances have been filtered out of the blood. And so at this point, you are reabsorbing them. You're taking them back, putting them back into blood again. Because initially, substances that get filtered out of the blood, it's almost, and we'll talk about this in lab, it's almost everything within blood. A lot of stuff, and it's stuff that you need, that your body wants to keep, initially gets filtered out of the blood. And that happens right here. But very quickly, so right in this very first part of the tubule right here, you are going to reabsorb what you need. So for instance, initially, all of the water from your blood gets filtered out, and you need that in your blood. That's what maintains your blood volume. So in this area, you will reabsorb a lot. You're going to reabsorb a lot of the water and reabsorb a lot of other substances too. So it's in this area that about 99% of all the reabsorption happens, and the microvilli are helping with that process. Okay, The next part of the tubule is this right here called the loop of Henle. So loop of Henle has a descending limb where the fluid is traveling down, and then it curves around, and it has an ascending limb where the fluid is ascending. It's traveling up, goes up the other side. And it is in this area where this area helps determine urine concentration. So how concentrated urine is going to end up being. And next we have in the purple area, the distal convoluted tubule, DCT. And just like the PCT, this is simple cuboidal epithelium, just little cube shaped cells. And there are two modifications here. There are two special types of cells here that have a specific function. There are principal cells, which help maintain the body's water and sodium balance. Okay, they help maintain the water and sodium balance. The other cells are called intercalated cells, and these help maintain the acid-base balance of the blood. Okay, and then the final portion of the renal tubule is called the collecting duct in the red circle. And this is pretty much exactly the same in terms of the cell types as the DCT. So same types of cells, same specialized cells. It still has principal cells. You have the intercalated cells here too. So this extends all the way down, and this is the longest part of the renal tubule. This goes all the way down into the pyramid. You guys remember the triangular structures that are like towards the middle of the kidney? So this extends all the way down to the tip of the renal pyramid, which, remember, I told you there's a little opening at the end of each pyramid called the renal papilla. It's a little like a hole. So this extends all the way down to that. And the fluid is obviously traveling down the collecting duct. And from here, it's going to go through that little opening to get into, remember, then you have this series of channels within the kidney that ultimately carry urine into the ureter, which carries it down into the bladder. And then from there, it can be released outside the body. So this is going to lead down to the opening at the end of each pyramid. Okay. So next thing is that there are two main types, two groups of nephrons. And most of the nephrons that are in the kidney are cortical nephrons. So that means they are located in the cortex. Remember, the cortex is the outer portion of the kidney. So this picture is showing you just a little segment, like a little piece of a kidney. Remember, the cortex is the lighter outer region. So this area right here, shown in the lighter color, is the cortex. Here is a cortical nephron. You can see that almost all the nephron, with the exception of a little bit of the loop of Henle, and its collecting duct, which always has to extend all the way down to get to the papilla, the opening. The majority of this is in the cortex. But there is another type of nephron, and this is like the other 15% of nephrons are called juxtamedullary nephrons. And these are the ones that are a little bit further down or towards the middle of the kidney, and they extend more deeply into the medulla. which is the region of the kidney that's in the middle. And you can see the difference here. So you have the top part of the nephron is in the cortex, but you can see the entire loop of Henle, which in these nephrons is really long. It extends all the way down into the medulla. So these are longer, and almost all of them is located in the medulla, not the cortex. Okay. I am going to do this next part here because that is not, I don't have a lot of information on just these next couple of things in my recorded lectures, but then we're getting to some parts where I have a whole lecture for, so I will skip that so I'm not repeating myself. But this next section is not. So I am going to go over the two capillary beds that are associated with the nephrons. The first capillaries we've already talked about, and that's the glomerulus. So remember, the glomerulus is here. This is the mass of capillaries that is specialized for filtration. It's that very first part of the nephron. Okay, here is the glomerulus. So it's part of the renal corpuscle, the first part of the nephron, and it is specialized for filtration. There is another capillary bed associated with nephrons that I haven't mentioned yet, and that's the next one listed. These are called peritubular capillaries. These capillaries branch off of the efferent arteriole. Efferent arteriole is right here. And if you need spelling, you can look at that right here. E starts with an E. Efferent arteriole. And these capillaries wrap around the renal tubule. So you can see in purple on both of these pictures, the purple is the nephron. And most of the nephron is the tubule, that long, coiled in parts, coiled up tube. And then the loop of Henle you can see. What's shown in red is the peritubular capillaries. So you can see they just kind of cover, like they wrap around the tubule. And especially they wrap around that loop of Henle. And if you look in your outline, you have a little bit of a description already written about these capillaries. They are very porous, so their walls have a lot of openings, which allows them to absorb solutes, so solid particles like ions and nutrients, and also water from the cells that make up the renal tubule. Okay, so basically when substances are being reabsorbed and they are coming out of the filtrate, they are having to pass through the walls of the renal tubule, but then immediately they're going to go into these capillaries so that they go right back into the bloodstream. Okay, so substances being reabsorbed from the filtrate are moving out of the renal tubule, which is shown in purple, and then moving right into this capillary bed. So that's why these capillaries are so closely wrapped around the renal tubule, because they have to be in close proximity to it to be able to reabsorb the nutrients and the ions and the water that are leaving the filtrate. They are like right next to each other so that reabsorption can happen really quickly and easily. Okay. So these are the peritubular capillaries. In the juxtamedullary nephrons, which here's one of those, these are, remember, the nephrons that are a lot longer. They have that really long loop of Henle that extends down really far into the renal medulla. The peritubular capillaries in these juxtamedullary nephrons, they do the same thing. They really are the same thing. They just have a special name when they are on these particular types of nephrons. They are called, it's called the vasa recta. So it's the same thing as the peritubular capillaries. It just is a special name for those capillaries when it's found on these really big juxtamedullary nephrons. Okay, and you guys have these two pictures in your outline, so that might be helpful just to be able to visualize these capillaries and see where they are. Okay, so one last thing, and then we're getting into the stuff that I do have a lecture, a recorded lecture on. So I think once I go through these last two definitions, I'll give you guys a break, and then we'll come back to a different section that I don't have a recording for. So the next two things here are the juxtaglomerular complex. And this juxtaglomerular complex, we have all these crazy names with the nephron and the urinary system. So the juxtaglomerular complex is located right where the distal convoluted tubule, which is here, it actually touches at a certain spot. It touches the afferent arteriole, which is bringing blood into the nephron. And right where they touch, there are two modifications to the cells that are directly touching each other. And so I'm just going to give you a quick definition or description of those two special types of cells. The first one is called, right here, macula densa cells. These are very tightly packed epithelial cells. That monitor, and actually that what they do is written right here in the PowerPoint. They monitor the ion concentration in the filtrate. Okay, so they monitor the amount of ions that are in the filtrate. The other cells, the granular cells, these are smooth muscle cells. That monitor blood pressure in the afferent arteriole. They monitor the blood pressure in the afferent arteriole. Okay. So this next section where it talks about mechanisms of urine formation, you know what, let me just give you, this is actually in the webpage that I've created that has the recorded lectures in it. But I'll give you, just for your understanding, like for the rest of the lecture, let me give you a quick description of those three things. And then we'll take a break and continue. So these are the processes that are involved in ultimately forming urine. The first thing that has to happen is glomerular filtration, which is when the kidneys filter blood to produce filtrate. Sorry, my cat is climbing all over the place. Okay, so kidneys are filtering blood to produce filtrate. That's the first thing that happens. And then you will have reabsorption. We talked a little bit about that. Reabsorption is when water and solutes, and remember, solutes are solid particles, things like ions and nutrients. Nutrients would be things like glucose and amino acids. Okay, so reabsorption is when water and solutes are reabsorbed from the filtrate back into the blood. And then there's one more process, and we haven't really talked about this one yet. It's called secretion. And this is when wastes or excess ions, or really any substance that is in excess, are secreted from the blood into the filtrate. Okay, so substances are moving directly from the blood into the filtrate. And these would be things that you want to get rid of, like you want to put them in the filtrate because you want to excrete them from the body. So once these three processes have occurred, everything that is remaining in the filtrate will be excreted as urine. It's going to end up becoming urine, and it's going to be, and it's going to be excreted from the body. All right, so we've come to a good place to maybe take like a 5-minute break if you guys want to need to do anything for a couple minutes. It is it's 10:21. Why don't we come back at 10:27 approximately, and then I will continue with some more stuff like that's a little bit further along in the outline. Okay, it is 10:27, and I don't want to keep you guys for too long, so I will get started if you are back. If you're following along in the outline, the next section where it gets into glomerular filtration, these definitions of like the parts of the filtration membrane, these are directly in the web page. If you look at the very first Unit 5 content folder, and if you go to that web page that I've created with like all the written content and diagrams and embedded videos and recorded lectures, those definitions are right in there. I will maybe just really briefly discuss what's in Filtrate. And I will give you a quick definition for the filtration pressures as well. This is a lot of what you guys saw though in the urinary physiology lab that you did last week. That was really helpful. I know it was a little bit early before we actually got to the urinary system, but it is a really helpful lab because it has a lot of the physiology in there, helps you with your understanding of that. So anyway, I am skipping the filtration membrane definitions because they're word for word in there in the web page. But in terms of the filtrate, I just want to be clear on what filtrate does contain. Basically, and you have this definition in your outline, the fluid that ends up being produced after blood is filtered is called filtrate. And it contains everything that was in blood except, and this is what you might want to add here, except blood cells and almost all proteins. those things are generally just too large to fit through the small holes of the filtration membrane, or remember those filtration slits. They're too big to fit through. So simply because of their size alone, they can't get through the filtration membrane. So all of the blood cells and almost all of the proteins, maybe a few really tiny ones can fit through, but for the most part, proteins are pretty big and they can't get through the filtration membrane either. What does end up going into Filtrate, you guys have a ton of things listed here. So it is a lot of water, glucose and amino acids. Those are the main nutrients that get into the Filtrate. Some byproducts of metabolism like lactic acid and creatinine. Urea is another metabolic byproduct of breaking down proteins. And then a bunch of ions end up in the Filtrate. So let me give you some definitions for the filtration pressures, which you did look at in the lab, but we'll go through those. Let me see. Here we go. Okay. So the net filtration pressure is actually the pressure that is responsible for creating the filtrate. And it involves two main forces. There are outward pressures, which are pushing fluid out of the blood. And there is an inward pressure, which is actually going against that pressure and pushing in the opposite direction. So the outward pressures that promote filtration formation or filtrate formation are just one, and it's the hydrostatic pressure, or glomerular hydrostatic pressure. And basically this is just blood pressure within the glomerulus. This is simply just blood pressure within the glomerulus. So that is the pressure that is pushing blood like across the filtration membrane, and it's promoting filtrate formation. If you look at the amount of pressure, it's about 55 millimeters of mercury. That is the largest pressure. There are some opposing pressures, but fortunately they aren't as big as this one. So this one wins and filtrate continues to be produced unless something is wrong, something there's an abnormality or there's a disease or something that's impacting these pressures, and then it can affect the balance. But normally in a healthy individual, the hydrostatic pressure, the blood pressure is the greatest, and filtrate will continuously be produced within the kidneys, which is good. That's what you want. So these opposing pressures, which actually push the opposite direction, are first of all, hydrostatic pressure within the capsular space. The capsular space is here, shown in yellow. And this is the pressure that's exerted by filtrate that has already been formed. So this is the pressure exerted by filtrate that is already here, that has already been produced. Okay, and that is a pretty low pressure, 15 millimeters of mercury. The other one is called colloid osmotic pressure. This is the pressure exerted by proteins in blood. And that pressure is about 30 millimeters of mercury. So even with that one plus the other pressure that's opposing, so 30 plus 15 is 45, that is still less than blood pressure, which is promoting filtrate formation. Okay, so that's good. You always want those numbers to be less than blood pressure within the glomerulus. Otherwise, filtration will not happen. And can you think of like what would happen if you can no longer filter blood? What if, go ahead. I mean,
dialysis.
Right. So a couple things, like, yeah, you guys are both right. So if your kidneys are not filtering blood, then all these like excess substances are just going to start building up in blood. And I mean, that it might seem like a no big deal if, you have extra ions that are building up in blood, but think about like a couple things. If you have too much water building up in your blood, that's going to add to your blood volume. And if blood volume starts increasing, then blood pressure is going to start increasing, and that could get way too high. And I mean, yeah, that could kill you. Or another thing is hydrogen ions. Hydrogen ions are acidic. So if hydrogen ions start building up in the blood, it's going to create an acidic condition. It's going to lower blood's pH. And if you can't regulate that with other mechanisms, I mean, one main way that we get rid of hydrogen ions is through urine. So if you can't get rid of them and your buffer systems can't keep up, then blood pH is going to drop, and that can kill you. So if that is happening, if the kidneys aren't functioning properly, yes, we do have options. You can go on dialysis where you have an artificial filtration membrane that is basically filtering your blood for you because your kidneys can't do it. So yeah, all of that is not good. So normally, if everything's normal, if you're healthy, that first pressure, the hydrostatic pressure, is the largest pressure, and that is going to ensure that blood is continuously being pushed across the filtration membrane. So filtration is happening, and substances do get removed from the blood. So that's what you always want to see. All right, so the next part, I have a whole recorded lecture for. Please watch it, because this is an important physiology piece of the urinary system. So it's like how your kidneys can regulate their filtration rate. And also this does influence blood pressure, because blood volume, basically filtration impacts blood volume, which directly impacts blood pressure. So in that recorded lecture, I described the two main mechanisms that are involved in basically regulating the filtration rate. So it's where you see intrinsic controls, renal auto-regulation. I discuss all of that. And then I talk about the extrinsic controls. So things that are not within the kidneys, but other mechanisms outside of the kidneys, like the nervous system and various hormones, how they can influence blood pressure by influencing filtration. So please make sure you watch that. I don't want to do the whole thing right now because I've already done it for you. I'd rather get to other stuff that you don't have a recorded lecture for. So that covers that whole intrinsic mechanism, extrinsic controls, all of the hormones that are there. So I am going to go to, so basically it covers the rest of filtration, that whole section on filtration. I'm going to skip to where it says step 2, tubular reabsorption. That is, let me see. 1, 2, that's like 3 pages past where we just were in defining those filtration pressures. Okay. All right. You guys see where we are? Tubular reabsorption. So I'll do a little bit of that because I don't have a recorded lecture on this part. This is a little bit easier. So we've talked about how reabsorption is just where your body is reabsorbing some of the substances that initially went into the filtrate. And there's a lot of information already in your outline, some kind of interesting facts. Our entire blood plasma volume, so blood plasma is the liquid part of your blood, which is mostly water. So your entire blood plasma volume is filtered out of your blood and it goes into your nephrons about every 20 minutes. So this filtration is constantly happening, and your entire blood volume passes through your filtration membrane in your kidneys every 20 minutes, which is kind of like crazy to me. That's a lot of blood going through very quickly. But what's also in here, which I've also tried to explain a little bit, is that almost all of that ends up getting returned to blood. So a lot of what is initially filtered out of the blood is going to be reabsorbed right back into the blood. So that is through this process, reabsorption. Basically, there's active transport mechanisms which use energy. They use ATP to pump substances back into the blood to force them back across. And there are other transport mechanisms that don't require energy. They just involve the movement of ions, going down their concentration gradient. Their natural movement would be to go from where there's more of them to where there's less. So they would naturally just move from the filtrate into the blood, because they all got filtered out of the blood and into the filtrate. So now you have a ton of these ions there and none in the blood. So they're just going to naturally move back into the blood. There is what's called a transport maximum for just about every substance that gets reabsorbed that has to use a transporter protein to get back into the blood. And that definition for the transport maximum is that this is a measure of the maximum amount of a nutrient that can be reabsorbed in 1 minute. So the maximum amount of a nutrient that can be reabsorbed in 1 minute. So anything that requires a protein, so if it's large enough, it needs a transporter protein to help it across, to help it move out of the renal tubule and into the blood. And these transport proteins can only carry one molecule at a time. So they can only work so fast. They can't carry 5 at a time. So basically they get maxed out. They can only carry so much and they can only work so fast. So that is basically the transport mechanism, the maximum amount that they can transport in 1 minute. So I'm just going to go through the major substances that are in the filtrate and how, the amounts and how and where they get reabsorbed. Sodium ions are the first ones we'll talk about because these are the most abundant ion in the filtrate, and these get reabsorbed first. They are the first thing to go. There is a sodium potassium pump that transports sodium out of the filtrate. It uses energy, it uses ATP, and it actively pumps sodium back into the blood. After that, other solutes, so other ions like chloride ions, calcium, potassium, these get transported out of the filtrate. And a lot of these get transported at the same time as sodium. And these other solutes, so I just listed a bunch, but these other solutes include things like other ions. They include nutrients like glucose and amino acids, and also vitamins too. Okay, and then lastly, so those are the main solutes that get reabsorbed: sodium, and then all the other ions and nutrients. And then water is another big thing. So the reabsorption of water happens in one of two ways. There is obligatory water reabsorption, and this is dependent upon the movement of sodium. So you'll see this like all the time whenever you have sodium within a cell or within a body fluid. If sodium moves from one place to another, water follows. Water follows the movement of sodium. So as soon as you start actively pumping sodium back into the blood, which is the first thing that happens, water is just automatically going to follow. It's going to be pulled along. So that's called obligatory water reabsorption. And that is actually how probably about 90% of water gets reabsorbed. Most of it happens that way. Okay, so that is automatic. Like your body doesn't control that. That is naturally going to happen no matter what. Then there's another type of water reabsorption that may or may not happen. It's called facultative water reabsorption. And this is the reabsorption of water dependent upon hormones. So this is water reabsorption that occurs due to hormones. Okay, so there are various hormones. I listed a couple of examples here, and these hormones are discussed. I know I didn't specifically talk about them today, but these are discussed in my recorded lecture. So these hormones would cause water to be reabsorbed if you needed additional water reabsorption. So like I said, water is automatically going to be reabsorbed when sodium gets reabsorbed. But then if you need even more water to be reabsorbed, it just depends on the body's needs at the time. If you need even more water to move back into the blood, then your body is going to release potentially one of these hormones, which is going to cause additional water to be reabsorbed. So that may or may not happen. It just depends on if your body needs it to. But just know that obligatory water reabsorption, that is always going to happen no matter what. You can't control that. Even if you don't want it to happen, it's going to happen. Okay, so the facultative water reabsorption is the only type that is a choice. Okay, so a couple other things. Now we'll go through the parts of the tubule and just talk about what gets reabsorbed. Remember that we said this when we initially talked about parts of the nephron. It is in the PCT, that's the very first part of the nephron, that almost all reabsorption happens. So almost all of it is happening there, but then there is a little bit in a couple other places. So 100%, you guys see some percentages listed there under the PCT. So 100% of glucose, amino acids, and lactic acid are reabsorbed here. Okay, and then about 80 to 90% of bicarbonate ions. So bicarbonate ions are HCO3, little subscript 3, HCO3, that's bicarbonate ions. So most of them get reabsorbed, 80 to 90%. Then about 65% of water, sodium, and potassium are absorbed or reabsorbed. and then 50% of chloride ions and urea. Urea is it's a metabolic byproduct of when your body breaks down protein. It is just a byproduct of that happening. So that.
Sorry, can you please repeat 65%?
Yeah, the 65% was water, sodium, and potassium. Okay, so all of that is right here in the PCT. That is where almost everything gets reabsorbed. You can see everything getting reabsorbed here. Then we go down to the loop of Henle, and you can see it is a little different in the descending limb compared to the ascending limb. Can you guys see what gets reabsorbed in the descending limb? Water. Yep. So it's about 15% of water gets reabsorbed here. We already had a bunch of water reabsorption in the PCT, but now you're going to have even more here. And then on the ascending side, in the ascending limb, you'll have reabsorption of about 30% of sodium and chloride. And also, it's not pictured here, but also potassium ions. So it's sodium, chloride, and potassium ions. So it's different. On the one side of the loop, you have reabsorption of water. On the other side of the loop, you have reabsorption of ions. Okay, so that, all of that we talked about so far is always going to happen no matter what. But then you can have some reabsorption in the last two parts, the DCT and the collecting duct. But this depends on the body's needs at the time. So here, these areas would be acted upon by the three different hormones: aldosterone, antidiuretic hormone, and atrial natriuretic peptide. So these would be secreted. And I don't want to take a lot of time to get into these because I have these, for these three hormones listed, I have the recorded lecture explaining exactly what they do. But basically, if those hormones are secreted, they're going to influence more water reabsorption. But these, it just depends on your body's needs. So if you don't need any more water reabsorption, then they just don't get secreted and nothing happens in these last two sections of the tubule. There just wouldn't be any reabsorption here. Okay, so because we're limited in time, I'm going to take like maybe 10 or 15 minutes to skip to, this is going to be in the second outline. And let me see. I want to talk about a little bit of urine composition. So the second outline talks about tests of kidney function. That is renal clearance. And I have a very specific description of that written in the webpage. So you guys can fill this exactly in from what's written there. But what I do want to talk about is the urinalysis section of that outline. And then I also want to mention chronic renal disease because I think I have an extra credit question about that. Okay, so urinalysis section, you guys find that? Let me find the right slide.
On page 4.
Thank you. Because I know my page numbers are different than yours, so I can't give you a page number. So thank you. Page 4, you'll have the urinalysis section. There is a little bit of information about this in the webpage that I created, but not in this much detail. So I want to go through this real quick. And then maybe I can I'll definitely do chronic renal disease, because I think there is an extra credit question about that. And then maybe I can do a tiny bit on body fluids. So urinalysis is basically looking at the physical and chemical properties of urine, and it can help determine if someone is suffering from a health issue or a certain disease. That's why it's performed. So normally the typical volume of urine that's produced is 1 to 2 liters per day. That obviously varies a lot depending on fluid intake, your diet, temperature, even amount of fluid loss through sweat that's happening. But generally it's between 1 and 2 liters per day. The odor should be non-offensive. For color and transparency, color, first of all, fresh urine should be clear, and then color should be anywhere from like a pale to deep yellow. You can actually see like a color range here. Lighter colors would indicate if you're more hydrated. So urine is more dilute because it contains a greater water concentration. And then if you're dehydrated, it would be a darker color because it would have less water. The yellow color, though, is due to urochrome. And urochrome is a pigment produced during the breakdown of hemoglobin. And it is just like a harmless byproduct of breaking down hemoglobin. And it's just added to urine so that it can be excreted from the body. If you have more concentrated urine, and this is what we were just talking about, if it's more concentrated, so more on this end of the scale, it's going to be a darker color. If it's more dilute, that would be on this end of the scale, so it would have a lighter color. Normal pH for urine is, so a wide range would be like 4.5 to 8. Sometimes it usually falls within a smaller range, like 5 to 7.5. The average number is right around 6, basically. So pH is a little bit acidic, and that does depend a lot on your diet, and our diets do tend to be acidic, which is a big reason why urine tends to be acidic. If you have increased protein in your diet, what's the immediate breakdown product of protein if you're breaking it into its smaller components? You guys remember what are the individual components of proteins?
Amino acids.
Right, amino acids. So if you have increased protein in your diet, so you're breaking down protein into amino acids, what is that going to do to the pH, you think, of urine?
More acidity.
Yeah. So if you're producing more acids because you're eating more protein, you're producing more acids as a result, then you're going to have more acid in your body. So you're going to have to excrete it through urine. So urine would tend to be more acidic. It would have a lower pH. If you have a vegetarian diet, then it tends to be less acidic, maybe more on the alkaline side. So a lot of it does depend on your diet. Specific gravity, we talked about this we talked about some of these, this included in the lab this week, and we will next week. Specific gravity is the weight of a volume of urine compared to the weight of an equal volume.
Can you say from the beginning, please?
the weight of a volume of urine compared to an equal volume, or I'm sorry, compared to the weight of an equal volume of water. So if you have 10 milliliters of urine, 10 milliliters of water, and you're comparing them, which one do you think would be heavier? Which one would weigh more?
Urine.
Yeah. So think about that. Urine basically is water, but then with added substances in it, like certain nutrients potentially, definitely ions, urea. So it's a little bit heavier. I mean, not a lot. We're not talking pounds here, but it's a little bit heavier than an equal volume of water. So water is given a specific gravity of 1. So if urine is a little heavier, it's going to have a specific gravity of a little higher than 1. The normal range for the specific gravity of urine is 1.001 to 1.035. And if there's a higher specific gravity, that just indicates that urine is more concentrated. There's a higher concentration of solutes in urine. Okay, and now getting into this slide, the chemical composition of urine, 95% is made of water. The other 5% are solutes. And these are the primary solutes that are found in urine. There's organic molecules, which are a little bit bigger, and there are ions. The commonly found organic molecules are the ones that are circled here, and they're listed in your outline. Urea is the first one. Urea, I've mentioned this a couple of times. It comes from the breakdown of protein, or specifically it comes from the breakdown of amino acids, which are made from protein. Okay, and another one is uric acid. That comes from the breakdown of nucleic acid. So when your body is breaking down nucleic acids, which are DNA and RNA, this is a byproduct of that. And then creatinine comes from the breakdown of creatine phosphate. And hopefully that molecule sounds a little bit familiar. We talked about that in the last unit when we discussed metabolism. And I mentioned creatine phosphate is a molecule that can be stored in muscles, but you can't store a whole lot of it in your muscles. And it can be easily used for ATP. And it's the very first thing that's used to make ATP, but you have so little of it that you end up not being able to make a lot of ATP from it. So then you have to use something else, which is glucose that you're typically using. Okay, so those are the organic molecules in urine, and then just a bunch of ions. They're listed here: sodium, potassium, chloride, bicarbonate. Those are the most common ones. Okay, I'll do this section, substances that should not be present. And we will talk about some of this in lab, I don't think not every single one. First of all, glucose. If glucose is in urine, it's called glycosuria. So if you had lab, you should hopefully remember this. Do you guys remember what condition, if you're suffering from what condition that would cause glucose to be found in urine? Anyone?
Diabetes?
Yeah, diabetes. So if you have uncontrolled diabetes, you're not treating it, you would often see glucose in urine. And I already explained that to the lab group this week. I'll explain this next week to you guys so you won't miss anything if you have not had lab yet. Proteins. So you shouldn't have proteins coming out in urine unless it's a very, very, very small amount. If you do, it can indicate hypertension. Hypertension is high blood pressure. So if you have chronically high blood pressure, so again, we'll talk about more details about reasons why when we meet for lab. Those of you that already met, you guys, we've already talked about this. Ketone bodies, these should not be found in urine. If you do see them in urine, it can indicate that someone is starving. It indicates starvation because they are using fat as their primary energy source, which is not normally the case. It could also indicate like a very, very low carb diet. And you would have had to been on that diet for like a while. It wouldn't just happen from like a day or two. Bile pigments like bilirubin, they should not be found in urine. If they are, remember bile is produced by the liver. So if bilirubin is found in urine, it can indicate a liver disease. Okay, also erythrocytes and leukocytes. These are blood cells. Erythrocytes are red blood cells. Leukocytes are white blood cells. Neither one of these should normally be found in urine. If you're seeing red blood cells, basically that indicates that there's bleeding somewhere in the urinary tract or in the kidneys. So that could just indicate like physical trauma. And often that can happen from a kidney stone. If someone is suffering from a kidney stone, it can cause physical trauma to the kidney, and then blood in urine is a primary symptom of that. Leukocytes, again, they shouldn't be found in urine normally. If you see that, it could indicate a urinary tract infection. Okay, so let me do chronic renal disease. And then actually we are running out of time. So I'm gonna stop there because the next section is just anatomy of like ureters, bladder, and urethra. And that is very simple. And then for body fluids, one of the main things there is the different types of electrolytes and their roles in the body. And if you look at the webpage that I made in the second Unit 5 folder, there is a chart there with each one of the ions, each one of the electrolytes, and everything that you need to be able to fill this part in is in that chart. So the function of the electrolyte and everything, disorders associated with them, everything that's in the outline is in that chart. It's very easy to read, so use that to fill that part in, and then you'll have most of that section done. So I'll give you this last little bit because I believe there's extra credit related to this topic. So chronic renal disease is when there's a glomerular filtration rate, or just filtration rate, less than 60 milliliters per minute. And just for reference, the normal filtration rate should be higher than 70 milliliters per minute. So if this rate has dropped to less than 60, that is considered chronic renal disease. If the filtration rate has decreased, then that means formation of filtrate is going to slowly decrease. And we talked about this a little bit ago when we were discussing filtration. What is going to happen if your filtration rate is decreasing? Now, it's not totally stopping necessarily, but it's greatly decreased. What is that going to lead to? What's going to start happening?
Will it affect the blood pressure?
It can definitely affect blood pressure, and just also waste products start to build up in the blood. And you're right. One thing that could start to build up in the blood is water, too much water, which can affect blood pressure. So that's a big thing. The other main thing, I mean, when some wastes build up in the blood, there are other ways of getting rid of them. It just depends. But another one that can be particularly dangerous is hydrogen ions. Do you remember why? What do hydrogen ions impact?
Is it the acidity of the blood?
Yeah. Hydrogen ions, whenever they start building up in any kind of body fluid, they are acidic. So they will drive the pH down. Yeah, so that's dangerous. If hydrogen ions especially start building up in the blood, it can lead to a decreased pH and it can cause acidosis. So there's a lot of bad things that can happen from a decreased filtration rate. This is now not always, but two of the most common causes are diabetes and hypertension, which is chronic high blood pressure. So they are two things that they don't have to, but they can lead to renal disease. And then actual renal failure is defined by an individual having a filtration rate that's less than 15 milliliters per minute. So that's way less than normal. Normally, remember, it's over 70. So this is if it's less than 15. That's very low. And that's when it gets really dangerous. So at that point, probably even sooner, somebody would likely need dialysis to artificially filter their blood. Okay, so we got through a whole lot. I'm pretty happy with what we got through. The section in the middle that we didn't cover is completely covered in the recorded lectures. So just, I mean, you do what you want, but it really, really, really would be helpful if you go in and look at those. I know some of you guys don't use them, and if you can get by with that, that's fine. But they're really going to help you fill in that part of the outline. And then that is really what the test questions come from. So please just help yourself by taking a look at that, at those. There's two or three, I think, recorded lectures that are in the first Unit 5 folder. So when you can, take a look at those. They'll really help you with some of those hormones, regulation of filtration rate, and regulation of blood pressure too. All right, well, I'm going to stop sharing. That's really all I have for you guys. If you have any questions, let me know. But if not, that's all I got for you today. I'll post the recording